guys, welcome to Bio 112 Online, your home for biology education. Today's topic, neurons and neuron functioning. This is part 4 of our series, Summation at the Axon Hillock. Up until this point, we've been describing the transmission of a signal along the length of a neuron from the axon hillock down the axon to the axon terminus. We then continued with our discussion of movement of that signal now across the synapse in the form of neurotransmitters, making their way to the dendrite of the postsynaptic neuron. Now that signal will have to move across the body of that postsynaptic neuron till it reaches the axon hillock of that postsynaptic neuron. Today's topic is going to explore what determines whether or not the action potential on that axon hillock of the postsynaptic neuron ends up firing. As a quick recap, in the presynaptic neuron at the axon hillock, we may have an influx of sodium ions. That influx of sodium ions will create a depolarization, allowing opening of the voltage-gated sodium channels, leading to a massive depolarization and a positive charge of the membrane. That will go through and open the voltage-gated potassium channels, leading to a hyperpolarization, and finally a refractory period. Now keep in mind that as those sodiums come in from one action potential, they seed the action potential of the membrane immediately adjacent to it. As a result, that action potential now moves as a wave down the length of that neuron until it reaches the axon terminus. Once it reaches the axon terminus, we then open voltage-gated calcium channels. The opening of those calcium voltage-gated channels leads to an influx of calcium, which will then activate the release of neurotransmitters. Those neurotransmitters will move across the synapse until they reach the receptors on the postsynaptic cell. Once they reach the receptors on the postsynaptic cell, in today's example, we'll be discussing the use of ligand-gated ion channels. As those neurotransmitters meet up with the receptors, they allow additional ions to enter the postsynaptic neuron across the neuron body to the axon hillock. There could be two main effects of this, though. The first of these is referred to as an excitatory postsynaptic potential. An excitatory postsynaptic potential is a synaptic signal that creates a depolarization in the postsynaptic cell. An example, this is glutamate. You may recognize glutamate as one of the 20 amino acids used in protein synthesis. One of its other main functions here is to be used as a neurotransmitter. It will move across the synapse and reach the AMPA receptor. The AMPA receptor is going to be a tetramer, i.e. it's going to be 4 proteins put together into a single unit. Recall that quaternary structure from our discussions of proteins. In this case, once those glutamate molecules hit the receptor, they will allow those 4 units of the protein to split apart, creating a pore, allowing sodium into the cell. So therefore, as we come through, release glutamate across the synapse, it reaches the AMPA receptor. As it reaches the AMPA receptor, you have the influx of sodium into the cell. That influx of sodium into the cell can create a depolarization. That depolarization can move across the dendrite, across the body of the neuron, until it reaches the axon hillock. Now this can have one of two main effects. It can go along and be sufficient to trigger the action potential at the axon hillock, or in some cases, it may be insufficient to do that. But we'll get to that in a moment. We have another main type of postsynaptic potential that can occur. That second type of postsynaptic potential is referred to as an inhibitory postsynaptic potential. An inhibitory postsynaptic potential is a synaptic signal that creates a hyperpolarization of the postsynaptic cell. An example of a neurotransmitter that does this is going to be GABA, otherwise known as gamma aminobutyric acid. GABA will travel across the synapse until it reaches the GABA A receptor. The GABA A receptor itself, again, is a tetramer. Once GABA binds to the GABA A receptor, those 4 components of the protein spread apart, creating a pore. As a result, allowing chloride ions now to enter the cell. As chloride enters the cell, it can go through and create a hyperpolarization. So viewing this now, the release of GABA across the synapse will activate the GABA A receptors. These GABA A receptors will allow chloride ions into the cell, creating a hyperpolarization because those chloride ions are going to be negatively charged. Now, with these 2 postsynaptic potentials in mind, we now have to go through and explore what happens to lead that axon hillock of the postsynaptic neuron to fire its action potential. This can happen in one of 3 main ways. The first of which, if the excitatory postsynaptic potential is high enough, then it will go through and trigger the action potential. If the excitatory postsynaptic potential alone is insufficient, we may still be able to trigger the action potential by getting multiple signals to hit at the same time. We refer to this as temporal summation, or the combined effects of postsynaptic potentials from the same synapse arriving in rapid succession at the axon hillock of the postsynaptic neuron. Temporal summation occurs when a single EPSP may be too small to trigger an action potential in the postsynaptic cell. However, if multiple signals from that same presynaptic neuron arrive at the axon hillock of the postsynaptic neuron at the same time, they may be able to build up enough of a depolarization to allow that postsynaptic neuron to fire its action potential. There we are. The other main way in which we might see these multiple postsynaptic potentials all arriving at the same time may be occurring in the case of spatial summation. Spatial summation isn't about a single presynaptic neuron, but is about the joint effects of multiple presynaptic neurons all affecting the same postsynaptic neuron. In this case, the combined effects of postsynaptic potentials from different synapses arriving at nearly the same time at the axon hillock may trigger the postsynaptic neuron. This can occur either as multiple neurons all sending EPSPs, but it can also start to take into account the idea of IPSPs, of inhibitory postsynaptic potentials. This is going to occur when the combined effects of both the EPSPs and the IPSPs together hit that axon hillock and determine whether or not the action potential will initiate in that postsynaptic neuron. So imagine for a second we have 3 neurons, 2 presynaptic neurons, one in red, one in green, and then 1 postsynaptic neuron in blue. We may run into a situation where the presynaptic neuron 1 fires an EPSP sufficient to activate the action potential. However, the presynaptic neuron 2 at the same time fires an IPSP. The combined effects of these would be that they annihilate each other, leading to no effect, or at very least may decrease the depolarization enough such that the postsynaptic neuron does not fire. In this case, presynaptic 2 essentially played a nope card and prevented that postsynaptic neuron from firing. The second way in which we might see this is if we have a presynaptic neuron number 1 that is firing a signal that is insufficient to trigger the action potential. However, the combined effect of a second EPSP from presynaptic neuron 2 may be enough to bump that depolarization high enough that the postsynaptic neuron is able to initiate its action potential. In this case, postsynaptic neuron only actually fires when both presynaptic neuron 1 and presynaptic neuron 2 send signals simultaneously. This complex interaction and communication between neurons is a critical part of our understanding of reflex loops and of higher nervous system functioning. But this is a topic for another video. So until then, stay safe, stay healthy, and I'll see you later.